Hello everyone. Today we are going to discuss about the topic cell culture, stem cells and along with that organ culture. So first of all, as an introduction, what is cell culture? Cell culture is the process by which prokaryotic, eukaryotic or plant cells are grown under controlled conditions. But in practice, it refers to the culturing of cells derived from animal cells. It was first successfully undertaken by a scientist named Rose Harrison in 1907. In 1885, Rogers for the first time maintained embryonic chick cells in a cell culture. This is the basic aspect or introduction to what is cell culture. Next, what are the substrates used for the growth of cells, or how artificial meats are required for the growth of a cell? And mainly there are three different substrates used in case of cell growth. First one, plastic. Disposable plastics are cheaper substrates and they are made up of polystyrene. They should be thrown at a proper place after the cell culture. Before that, it needs to be treated with gamma radiation or electric arc simply to develop charges on the surface of the substrate. In addition, other plastic materials such as used are Teflon, polytetrafluoroethylene, Thermomax, polyvinyl chloride, polycarbonate, etc. Second substrate is glass. It's an important substrate used in laboratory in several forms such as test tubes, cover slips, pipettes, flasks, rods, bottles, petri dishes, several apparatus, etc. These are sterilized by using chemicals, radiations, dry heat, using oven or moist heat in an autoclave. Third one, palladium. For the first time, palladium deposited on agarose was used as a substrate for the growth of fibroblast and glial cells. So these are the three different substrates. Now let's move on to equipments used for cell culture. First one and important one is laminar airflow. It's crucial to keep the cell culture working environment ultra clean and with no biological or particulate contamination. So in order to perform that, a laminar airflow hood is essential piece when working with cell culture. The downward flowing filtered air functions as an invisible curtain that reduces the chance of airborne contaminants from entering the workspace. If safety is required for the user, then class 1 or 2 or 3 biosafety cabinets should be considered. On the basis of this, there are three different types of hoods present in laminar airflow chambers. Second instrument is incubator. Cell cultures require strictly controlled environment in which to grow. Incubators enable the correct growth conditions by controlling the temperature, degree of humidity and carbon dioxide levels. Third one, an inverted microscope. Visualization of cells is required to monitor cell morphology and identify contamination. So, for better visualization, an inverted microscope is used. Third one is centrifuge. It is capable of generating centrifugal force for at least 200 into gram. Gravity is needed for pelleting suspended cells during various culture protocols. Third, next one, a water bath. Water bath set to 37 degrees Celsius commonly found in cell culture laboratories for pre-warming medium before use and thawing frozen cells and reagents. Fridge and a freezer requires the reagents need to be stored at 2 to 8 degrees Celsius for the purpose. A fridge or a freezer can be used. Next, a liquid nitrogen storage suitable for long-term storage of temperature below minus 130 degrees Celsius. Next, a hemocytometer used as a counting chamber device to count the cells after the culture. Next, pipettes, basic requirements, cell culture vessels and consumables. Supporting consumables for cell culture includes objects such as pipettes, tubes, etc. It should be sterilized before use. Second, now let's move on to the primary stage of a cell culture. Before culturing, a single cell need to be isolated from by the following processes. So the tissue sample is pressed through a 100 mm mesh and it is placed in a petri dish containing BSS or basal salt solution. The cell now it forms the cell plus tissue debris. The above process is again repeated, and the substance is passed through a 50 micrometer mesh and obtained more cells and plus tissue debris. Remove the above two processes and pass through a 20 mm mesh. Now we can obtain viable cells plus the tissue debris. The tissue debris is removed and the cells are counted using a hemocytometer, and the viable cells plus damaged cells are diluted to a medium of up to 10 raised to 4 cells per milliliter, and separate the viable cells from damaged cells. Incubation is done, and at last primary adherent cell culture is obtained. So this is the isolation, how a cell is isolated from a tissue sample. Two different processes are required. That's the basic process for cell isolation. With the help of two enzymes, also cells are isolated by first one using collagenase and by second one using trypsin. By collagenase, it uses biopsy method, that is a tissue sample is extracted or cut and the medium plus antibiotic forms a biopsed salt solution, tissue sample, and it is then passed to BSS antibiotic, and the tissue is dissected and we obtain a chopped tissue. They are washed by setting and complete medium plus collagenase incubated for 1 to 5 days. These disaggregated tissue plus tissue residues are obtained. It is pipetted, and the dispersed cells plus residual culture of epithelial cells are formed. It is then washed and cultured, and primary cell cultures are obtained in two different ways. This is the isolation of cell by the mechanism of collagenase enzyme. Next one, by using trypsin. Two different methods, cold trypsinization and warm water trypsinization. The tissue sample at first is extracted and chopped to 2 to 3 pieces, and it is washed more if required. In cold trypsinization, the substance or the tissue sample is transferred to a 25 ml glass vial and add BSS or basal salt solution and dissected, placed on ice, and vial cells tissues are obtained and they are soaked in cold trypsin for 1 to 6 hours. Next, the added trypsin is removed, disaggregated cells plus tissue residues are obtained. Next, it is incubated at 36.5 degrees Celsius for 20 to 30 minutes. 10 ml of medium with serum is added and pipetted to form dispersed cells. They then forms the cells for primary cell culture. Second method that comes under trypsinization, that is warm water trypsinization. In the same way, the tissue samples are transferred to a 250 ml flask. The tissue pieces plus 100 ml trypsin added to stirred for 3 to 4 hours, and pieces plus trypsin is obtained. The trypsin is removed, which we have seen before, and centrifuged, and the medium is added. Dispersed cells of the cells are dispersed in the medium. Count the cells using hemocytometer. Plating is done and incubated at 48 to 72 hours and can be used for primary cell culture. It's a basic simple procedure. Just by a glance we can get an outline about what is trypsinization. Two types, warm water trypsinization and cold trypsinization. So that's all about cell culture. I just briefed it. Next, what is organ culture? Organ culture can be done by various different types. That is, it's not a fully functional organ is fully cultured in the following procedure, but a tissue sample or a piece of the organ is cut and cultured. That's the way it is preserved. So first one, they are cultured on plasma clots. It's prepared by mixing 5 drops of the embryo extract with 15 drops of embryo plasma in a watch glass placed in a cotton wool pad. The cotton wool pad is put in a petri dish, and time to time cotton is moistened so that excessive evaporation should not occur. At last, this technique of organ tissue is placed into a raft of lens, paper, or rayon. The raft makes easy to transfer the tissue, and the excess fluid can also be removed. Second, culture on agar. Solidified culture medium with agar is also used for organ culture. Nutrient agar medium may or may not contain serum. When agar is used in the medium, no extra mechanical support is required. Agar does not allow to liquefy the support. The tumors obtained from adults fail to survive on agar media, whereas embryonic organs grow well. Third one, in liquid media, it consists of all the ingredients except agar. When liquid media are used, generally perforated metal gauge or cellulose acetate or a raft of lens paper is used. This possibility provides support for the culture of organ. Next, that comes under organ culture is whole embryo culture. The culturing of embryo in a suitable medium prepared is poured into watch glass, which are then placed on moist F7 cotton wool pad in petri dishes. In the same way, how the tissue or cell was cultured using excess basil salt solution, and the whole embryo is also cultured. Next one. So that's all about organ culture. Next, move on to the topic stem cells. What are stem cells? These are cells that have the capability of self-renewal and differentiation. Self-renewal is the ability of cells to proliferate without loss of differentiation potential and without undergoing senescence, that is aging, biological aging. A stem cell is a blank cell or precursor cell that can give rise to multiple tissue types such as skin, muscle, or nerve cell. A stem cell is essentially the building block of the human body. They comprise the following satisfactory needs, that is, they are totipotent, pluripotent, multipotent, and unipotent. Totipotent cells have the capability to produce all types of cells of the developing organism, including both embryonic and extra-embryonic tissues. Pluripotent cells can only make cells of the embryo proper, but make all cells of the embryo, including germ cells and cells from any of the germ layers. Therefore, they can make any cell of the body. Multipotent cells can only make cells within a given germ layer. For example, multipotent stem cells from a mesodermal tissue like the blood can make all the cells of the blood, but cannot make cells of a different germ layer such as neural cells or liver cells. Unipotent cells make cells of a single cell type. An example is a germ cell stem that makes the cells that mature to become egg or sperm, but not other cell types. So these are the basic structures or basic functions of a stem cell: totipotency, pluripotency, multipotency, and unipotency. So we have come to a conclusion about, we have discussed about organ culture, stem cells, and cell culture. So thank you.